Welcome to Catalyst Conversations. This is our first event of 2026, and each event just keeps getting better and better. We have about 5 to 6 events a year, so be sure to subscribe to our emails so you know about upcoming events. My name's Alberta Chu. I'm leading Catalyst Conversations. We're really glad you're here. We're glad you got out of the house, and we hope you stimulated your senses. Thank you to our speakers for all of the show and tell and presenting of your work with the plants and the music and the scents and everything. Thank you so much. So, Catalyst Conversations' mission is to support artists and scientists. We put them on a stage and they converge, connect, exchange ideas, and we all make new connections. So we're going to have a discussion with curator Laura Zitrain today. We'll learn all about the speakers and their work, Scooby, Jerry, Meg, and Christina. And then we will have a dialogue with the audience as well. So yeah, we have a fantastic program tonight. Our 4 speakers are creative, problem solvers, curious, artists and scientists, and artists and scientists all in one. Okay, Laura Zitrain.
Thanks so much, Alberta.
Skater.
Hi, everyone. I'm Laura Zitron. I am so happy to be here with all of you tonight in the middle of the woods, in the middle of winter, doing very nerdy things together. So thanks so much for being here. We have a great set of mini presentations coming up. And before we dive in, I was hoping that we could have a moment to introduce these amazing people up here today. In all of my years of curating, I have not quite come across a set of individuals like this before, where people are interested in pushing and remixing the boundaries of sensory experiences. Is it on now? It needed to be adjusted. And here we are. Speaking of sensory experiences, and each one of the speakers has prepared like 3 different images for our presentations. And before we jump in, if y'all could share a little bit about yourself, who you are. And I was curious, given how dynamic you are and how it's not just interesting art or cutting edge research or trying to invert what is music, but it is weird and it's delightful. And I'm curious if your 8th grade classmates could see in through time and see what you're up to today. Would they say, like, yeah, that was definitely Jerry, I could see that immediately? Or would they be like, my mind is blown, I have no idea where I am or who she is? So Scooby, go ahead first.
Hello, everyone. So my last name is Leposki. And in 8th grade science, my teacher called me the law because I got perfect scores on every test. Wow. But he also knew that I was an artist. So I feel like Mr. Goodrich, wherever you are, it's like it's a mix of all my interests as an 8th grader.
Yeah, 8th grade. I'm not sure about 8th grade, but 9th grade, I think, is kind of when I think things kind of settled. I had quite an inspirational high school biology teacher who, on the first day of the class, jumped on the desk and started howling like a gorilla and stuff like that. And that just really just drew me into it. And so I'm not going to jump on around and howl like a gorilla, but that was, I think, yeah.
Great. Can you hear me okay? Great. I just got the real answer because I recently went to my high school reunion and I hadn't been back since. And everyone was very surprised that I was a scientist. It kind of came out of the blue at the end of high school for me.
Ooh. It may not come as a surprise to anybody, but I was a huge nerd in 8th grade. And so I think I would have surprised myself in 8th grade to know that I was also doing art and on a panel like this. But I don't think, but it all makes sense, kind of like looking back and connecting the dots. And so I think, yeah, for everyone who thought I was a weird nerd, like that is still true. So.
Well, thanks so much, y'all. I mean, it's interesting how there is a mix of direct line to a slow emergence over time. Well, I'd love Scooby for you to take us off. And I'll ask you a little bit after your couple slides.
Actually, that question's a good lead into kind of my journey. It's like people always ask me like, how did you get started making music with plants? I mean, there was research and like experiments in the 70s, but really there's no precedent. So my 8th grade self probably knew I was going to be involved in some like art and science, but probably not making music with plants. So initially, actually, some folks had already asked me like how I got started, but I kind of learned about the technology just on a whim, like through a Kickstarter campaign. And I thought the technology could be useful in my practice as a sound designer. Like if I had writer's block, I could connect to my houseplants and maybe they could make a better melody than me, which is probably true. And so when it really got interesting and kind of the practice started taking off was when I got invited to the Isabella Stewart Garden Museum to perform in the courtyard. So like the image on the left is like the first performance, and then on the right is like the last time I performed there a couple of summers ago, and kind of had learned some things in that 5 or 6 year span. So this is actually at the Gardner as well. This is Rashid Johnson's installation, Antoine's Organ. I don't know if anyone saw that exhibition. It was called Presence of Plants. So all contemporary art that involved living plant matter. And I was part of a residency then, and so I asked them if I could come in on the day the museum was closed and record with that installation, because I just saw it as like a big instrument. So I connected to the plants and I kind of hopped inside of that structure. There was an upright piano, so I just kind of improvised on the piano based on what I heard the plants were doing. So really trying to push the practice, like where I can take the plant music. So these are some images from an ongoing public art project called Hidden Life Radio. I've been talking to folks in Cambridge where I live, and they said, have you ever connected to trees? And I hadn't, because I kind of just logistically, I'm like, wasn't sure how I was going to get up in the canopy, and also like how to power the equipment and things. So I got a grant like the year the pandemic happened, so that I had another year to figure it out, which was great. So that image with the solar panels, that's a system I designed that's pretty autonomous. So it just got installed in the canopy by Urban Forestry, and then it basically sonified the tree's activity from leaf out to leaf drop. So part of my interest, I guess the science part of it is like scaling it up. Like, okay, I've listened to plants, for an hour at a time. What if I listened to it for like 5 months? Like, what would that experience reveal? So, this was a copper beech tree, and actually Urban Forestry connected those electrodes to that single leaf, and it live streamed for 5 months without like any issue. Like the leaf didn't come off in a storm or anything. So that tree produced a live stream for like over 5 months.
And Scooby, could you take us through that last image? What exactly is happening here for the folks who haven't gotten a chance to talk to you one-on-one? It looks like there's an input with the leaf.
Sure.
And a power generation.
So the solar panels are really powering the device that translates the electrical activity into the musical information. And then inside a little Pelican case that's mounted underneath the panels, it's kind of beaming that data to the public library branch. And there's a computer that receives the data, then it turns that into music, and then it live streams it. I've had some people like reach out wanting me to do this type of thing, like in old growth trees in Australia that are like hundreds of feet, tall, where you have to climb up there, spend the night in those trees. But the issue though is really like I guess the technical issue of like how to power it. And you could parse the data and then send it. But for me, I try to keep all my projects like in real time. So the factors that you're experiencing, even the bromeliad out there, it's experiencing the same conditions that you are. So for me to like parse the data, then send it, that's not really where the approach I want to take. So this is just a shot of me in the Angeles forest on one of the ridge crests, like a lot of my work involves me going out to the middle of nowhere and connecting to the plants there. That's really where like my interest lies. So all those factors that created that moment with that particular plant, be it climate change, like different types of environmental situations. Sometimes they're geopolitical. I've been in the Middle East, and some of the plants have been kind of victims of those conflicts. So all those vectors interest me, and coming through the plant and what you hear through the plant is sort of like kind of a living witness to what those conditions are. Okay, thank you.
Thanks so much, Scooby. Now, Jerry, I've been told that you may or may not be that foot on screen.
No. But it's would be interesting if they were my foot. So yeah, I'm a neuroscientist by training, and this Jimmy Fallon image I think kind of captures what I'm interested in studying. Which again maybe the eighth grade or ninth grade of me wasn't prepared to be to be thinking about this, but essentially what you see here is this game called What's in the Box. I mean, maybe if you guys have played this game at some level where you might put something in a box and you're kind of there trying to guess what it is by your sense of touch. And so I work on touch, somatosensation. And when you think about this kind of a game, there's a lot of things happening in your brain that's that you're trying to figure out here, right? And so on the one hand, you hand your hand in a box, you're searching around, you're trying to find an object, and then you land upon an object, and then you're trying to figure out what is this I'm touching, right? And there's two things that are happening as in your brain that's kind of doing this. On the one hand, right, your touch receptors, whatever you're touching, those are going to be transformed into electrical signals that get to the brain, right? But on the other hand, what you're also trying to do is trying to figure out, well, what is it that I'm touching? You're trying to assign some sort of meaning to the object, right? And so how do you know that what you're touching is a foot versus a grape or whatever, right? And so there's this aspect of your experiences, your prior learning, your prior, as you move around the world, that you've built up experiences that are then used to then interpret what you're sensing. So there's this interface between what people refer to as a bottom up and then this interaction, what's referred to as a top down. And so I don't study Jimmy Fallon, but I do study this question in mice. And mice, for those of you not aware, really have an acute sense of touch using their whiskers, right? So they feel their way around their environment using their whiskers. And so forces touch the whiskers, and that deflects sensory nerves on the follicle of the whiskers, and that information gets transmitted up to the brain of a mouse. And so we can essentially teach mice to play these types of what's in the box games and ask, well, how are they interpreting these signals, and what is going on, at a very circuit mechanistic level in terms of how this information is being processed. And with mice, we have access to a lot of interesting tools and abilities to really understand, how electrical signals are being communicated, how these circuits are organized. And this image over here on the left and right are images. The left is images of real neurons in the brain of a mouse, and on the right is an artist rendition of some of the studies that we've been doing, where we've been trying to look at how neurons are connected. What kind of molecules are shaping the patterns of the activity in the neurons, and we're specifically studying the part of the brain where at this interface where we are trying to process this bottom-up information and then interpret that through this top-down information that we've learned through experience. So that's what we do in the lab.
Wonderful. Thank you. I was just wondering if we could play some of the plant music for the mice, how they might, how that would change in your experimental setup when they're doing what's in the box. If they would have an aversion to the bromeliads, singing its song.
I'd be happy to invite Scooby to our lab and try some stuff out for sure.
They might start doing yoga though.
Yeah, it might calm them down.
Yeah. Well, from mice to mosquitoes.
Hi. Can you hear me again? Oh, how about now? Hey.
Journey's white.
Yeah, let's use that one.
Yeah. Okay. Is this on?
I think that's on.
I promise I'm a scientist, even though I can't use microphones. Pretty good at microscopes. So yeah, I going back to my 8th grade self, definitely thought I was going to go to art school. And then I got to college and I learned that neuroscience was a major and there was no turning back. I think I'm here today because I'm kind of obsessed with smell and the chemical world. I love scent in all of its forms. And in my work, that's taken the study of this lovely creature here. This is Aedes aegypti mosquito. And so I study mosquitoes because they're the deadliest animal on the planet, even more so than people. So, this is the number of deaths per year by organism. What to do about humans, super hard problem to solve. But stopping mosquitoes from finding and biting people is something I think can be done during my lifetime. And I think to do this, we really need to understand more about their sense of smell because they rely really heavily on smelling our body odor when they're trying to find us to bite. And body odor, wrong one. I don't know if you can see this person here. This is meant to be some of their body odor, but body odor is actually hundreds of different chemicals that are somehow telling a mosquito that this is their target and they're going to get a blood meal. And so what we do in my lab is we look at mosquito behavior. We try to identify different compounds on people's skin. And then we look at the sense of smell in the mosquitoes, starting at the antennae, which is sort of like their nose, and going into the brain to look at how different odors are processed by basically making different types of mosquitoes with neurons that we can see in these pathways and developing all sorts of methods to study how they respond to different smells. So yeah.
And so Meg, if one of the possible pathways to deflect mosquitoes, could it be by doing a pretend flower that doesn't exist anymore from Future Lab and then putting it over there and then driving them crazy as they circulate again and again and again? Or is it you're thinking the pathway to less mosquitoes biting less human is something different?
So, I mean, I think the pathway to stop mosquitoes from biting people can be either what you're saying, which is something that's so attractive it could outcompete humans. And
all their delicious blood scent that's coming off.
yeah, if you could make something that smells better than we smell,
or worse for them.
Yeah, probably worse to us. But the flip side would be some kind of repellent that lasts for many hours or days that people actually want to wear and isn't gross and like doesn't have a whole like history that people don't like. And I think we could do this,
Christina, from the perfume scent curious side.
Amazing. So, no worries. So books on my. I get it. I started. I'm a biochemist. and a synthetic biologist. I work in bioengineering. How do we work with biology as a technology to like do stuff with us and for us? And that has taken me to many different places. I was a writer and an artist and a marketer and a storyteller. These days, I run a small studio for kind of storytelling and biotechnology. We design lots of different things, whether it's websites or brands or products or experiences. And a lot of that question, actually, the woman in the center is my business partner now. But we all have long asked this question, like, can we make GMOs that we can love? Right? Like things that actually like what is it? What is the kind of things that are meaningful to us and exciting to us and beautiful? What are ways that this kind of beautiful technology that I love about living things that can do all these amazing things in the world? How can they be more sort of special and inspiring to us? So that has led me to a lot of different places in my life. One of the things that I'm actually best known for is making cheese out of bacteria that came from human skin. And so actually, one of the really interesting things about mosquitoes, those are ones that like bite your ankles, like and kind of they actually are attracted to like foot odor in particular, right? And you can trick them into biting cheese because there are certain kinds of cheese that make exactly the same odor that is in cheese, like cheese and skin, those same bacteria, like in your toes and in the cheese. Okay, and so.
Now we're on to a real product.
Yeah.
Everyone's excited to buy.
Yeah. And so I learned about this fact when I was a graduate student. And I was in the lab and I was working on a really interesting project funded by the NSF that put scientists and artists together. And we had like really like other than spend a month together, like no other brief. It was just like figure something out. And so the artist that I got paired with is a smell artist. And she is like very like amazing, like a long career, very famous smell artist. She shows up in the lab like, and I'm like, I'm a microbiologist. And she's like smelling everything, and she's making smells of body odors, and like painting them in the walls and galleries, and to like experience the smell. And so like, okay, let's look up what body odors are made out of. And I found this thing about mosquitoes and the feet and the smells. And so I was like, oh my god, well, I have feet, I have Q-tips. There's a grocery store across the street. Let's make some cheese. And so that's what we did. And so that project was really like, it was super interesting. So like, I mean, it became like this very big sort of scientific analysis of all of these bacteria, like which ones grow in the cheese, how do they make the different smells, and this like really cultural experience too. Like, why is it gross? Like, it is the same bacteria. Like what kind of drives our feeling and our about like when it's appropriate to have bacteria or others. And like for this kind of aesthetic question about bioengineering, right? And biotechnology, like cheese making is a kind of technology, right? Where we're actually like doing that thing of like, we are working with microbes and through microbes to transform something into something really tremendous and valuable, right? And so can we do technology like we do cheese making?
And did it taste like cheese? I imagine everyone's wondering.
It did taste like cheese. Yeah. It's
wonderful.
I got better at cheese making. So the foot one that I made in that, like, horrible. The worst thing, not good. But I did get better at it over time, and
Not that one.
It's great. But we say it's not cheese for eating; it's cheese for thinking. And so like that helped a lot.
That's a great brand name.
Yeah, exactly. Yeah. And maybe I'll talk about one other project to give you another flavor again. So I spent a long time working at a biotech company here in Boston called Ginkgo Bioworks. And one of the kinds of technologies we're working with is using yeast to produce molecules using fragrance. And so I was like, okay, we're talking to all these like the fragrance houses, and we're like, let's talk to your perfumers. And they're like, no, you talk to procurement. And like the chemists, like, go away. Like, and I was like, no. There's, again, technology can be creative. Like, we can be part of the brief to like make something like creative and beautiful. And so we're like, well, maybe we could make Jurassic Park for perfume, and like that would be interesting and creative. Like, could we do it? And that started actually like a 10-year-long journey for me, where we did all this kind of like trying to figure out, like, could we do it? Like, could we find specimens? Where could we be? This is in the Harvard herbarium. So each of these folders is like holding like a genus of plants, and like specimens that had been collected from around the world. And so we went and actually like sort of searched through these stacks, like they're not digitized.
This is a library, what
It's a
we're looking
library.
at. Yeah. Of dead plants.
Dead plants. We found a bunch that were in fact extinct. This is one. It's a kind of hibiscus. It's related to hibiscus. It's from Hawaii. When people brought cattle, like colonists came to Hawaii and brought cattle and sort of really changed the landscape of Maui. And this is one of the plants that went extinct. We've got a specimen of that DNA. We put that DNA into yeast the same way we did the other kinds of fragrance molecules, and we figured out what it smelled like, and worked with Sissel to produce an artwork based on that project and this science. And then the outside, you can smell the Future Society scents, which are kind of commercial, like fine fragrance, working again with we finally got to talk to the real perfumers, and like work with them to make something beautiful based on this kind of scientific story.
Thank you so much, Christina. So, Scooby, do you look at something like this and you're just like, if I could just put an electrode there and there?
So I think I'm good. Okay, I'm back.
Wow.
I had a performance at the Mass Art Museum, and they invited me. There was an artist who created incense from working with scientists, and it was the one incense stick was the first forest, and then the other incense was speculative, the last forest, which I think they determined it would be like in the Amazon. The first one was in Cairo, New York, right? That's where they discovered the oldest forest. So I was thinking like, okay, they wanted me to create some sort of sonic experience to accompany when they burn the incense towards the end of the evening. And so I was kind of doing some research to figure out like what plants could I use that are sort of like, like descendants of prehistoric plants. I know ginkgo is like a late cousin of prehistoric plants. And I think I used, actually I'm trying to think of the plants I used, but it was a fern, and there was like a palm tree that was like a direct descendant. So that was the closest I could get to like kind of connecting to a prehistoric plant.
Like the ancient music buried in those layers.
Yeah, I was sort of like kind of thinking of it as like a continuum, like for this plant, this is where it's at right now in time. And like what does it sound like? And then, if I was around another million years, like what would that plant's descendant sound like? Yeah.
And I'm curious how the mosquitoes might also interact with an unusual olfactory experience. I know when we're touching base before, when you're looking at an aversion or an attraction, how does that show up for you guys in the lab when you're studying? And is it just from the behavior, or is it from the brain imaging?
So when we're deciding if an odor is attractive or aversive to a mosquito, we really just look at their behavior. Do they move towards it or away from it?
Do they bite it repeatedly?
Yeah. Do they try to really get to it? I mean, if they're attracted to something, they're really going to try to get to it pretty ceaselessly, is probably everyone's experience, like in the summer, especially around here. Yeah, we do imaging in the brain, but it's usually separate from watching them and how they respond to different odors.
Mm-hmm. And what's some of the most unusual responses you've gotten to your work before? I imagine, Christina, we can figure that one out.
The cheese project was a very interesting one to see the response, because I got to see people reacting online, just sort of seeing the tweet or like the headline, and then I got to see people sort of interacting with the cheeses like in the context of like a workshop or gallery sort of space, and so like online it's like, did you do ball cheese like that? And a lot of that. But in person, like there was that maybe like that first sort of like aversion, but then people actually like always kind of like leaned back in and like we're asking a lot of questions. We're like, oh, well, what is that? Like, how is it in cheese? What is it on skin? How is it different? Like, and so it was really interesting to kind of see the curiosity kind of turn on live, versus again like the kind of like drive by gross out.
Mhm. Anyone want to add any unusual reactions you've
I don't
come
know
across?
unusual, but we published a paper a few years ago where we were talking about touch and how there's ways to keep the brain plastic through sensory experiences. And somehow we're Googling around and somehow the article made its way into a retirement home. There was like the retirement holder had a weekly publication that they, distributed to people living there, and our article made it there. So that was a little bit unexpected.
And how did you figure that out? Did you get a phone call one day?
No, we were just, I don't know, somebody sent it to me, and it just said this is, we were Googling around looking for articles related to what we were doing and saw it in some PDFs, that was in a retirement home. So you never know sometimes.
Wonderful. I thought you were going to end up in, so there would you be put fuzzballs on all the tables to have a.
Yeah, things to keep people's brain plastic and kind of nimble.
And do you see something your work connecting outside of the mouse realm into the human realm about plasticity and how what we know about central nervous system and touch?
Yeah, I mean, I think in general, like the brain is there's the brain's trying to strike a balance, right? Because there are certain things that we learn that we don't want to forget, right? And then there are things that having that ongoing plasticity is good for not only learning new things but also for repair, right? In case of you suffer a stroke or something like that, right? And so the things that we're studying, we're trying to kind of figure out that space, that parameter space, and see what is the optimal situation.
What does a good plasticity look like?
Once essentially we're finding that trade-off, right? Because as I mentioned, you don't want to take a lot of effort and forgetting the things that it's important to remember, but at the same time you do want to keep things kind of at a refresh and be able to kind of pick up new things. So it is a little bit of a balance that we're trying to.
Hmm. And speaking of balance, I know, Meg, in some of your work, I saw that comes up. You tend to look at where mosquitoes are going to bite, but also potentially to lay their eggs. Is that a different pathway? Are they deciding something different when they're looking for a safe place to lay their eggs? How do they make sense of their environment in that context?
Yeah. So the species of mosquitoes that we mostly work on, the Aedes aegypti, the Zika, yellow fever, dengue mosquito, when they're looking for a person to bite, so only females bite, and they're in this state of, we call it host seeking. All they need this blood meal and they need it to mature their eggs. But once they've had it, they go and hide out for a couple days and they change their state entirely. So they go from being awake during the day to being awake at night, and they start to look for fresh water sites to lay their eggs.
When we say look, do you mean smell it? What do you mean?
Yeah, I mean, so they are sensing humidity, and then they actually dip their legs in to try to determine, is the water pure enough for their eggs to survive, because their eggs won't survive in salt water. Whether they may look, we actually don't know if they're using their visual system or not.
But it sounds like there's something going on with their tactile element, or what's happening with their legs.
Yeah, so insects actually, they taste on their feet. So there
Back
is
to foot cheese. It all comes back to the foot cheese.
yeah, smell and taste are a little bit different in insects than they are in mammals, right? We have a mouth, and that is
Convenient.
we talk about food, like that's eating. Taste happens in the mouth. A lot of other animals, like flies and mosquitoes, they have, we call it contact chemosensation. So
Can you say that one more time?
contact chemosensation, like chemical sensing. So olfaction or smell would be volatile, non-contact chemosensation. And it's less about the appendage they use and more just like, is it something they can touch? Well, if they're touching it, it becomes taste if it's a chemical, not if it's a texture. And if it's something that's floating in the air, then it's smell. It's a little bit of a sort of the spectrum of chemical senses in other animals.
Fascinating. Well, I think we might start circulating a microphone around, or if anyone likes their hand. Okay. Maddie, do you have an extra mic? Or should I? Oh, here's Amber, another board member who's fabulous in all many different ways. Oh, okay, great.
All right, who's first?
Please.
Here, I think that's on. I'm married to a man who's really a foodie, and I noticed that he really likes these stinky cheeses that to me smell like feet. And I'm wondering if the bacteria in those cheeses is at all related to anything that you're doing. Like you made feet bacteria cheese, but doesn't that already exist, or is that something different?
So maybe there was it's a little hard here. I'm going to repeat the question and make sure that I understood. You're saying like if it's the same bacteria, like you're saying, but my bacteria cheese is the same. It's just normal cheese. Like what are you saying? It's not special. Is that what you're kind of the question?
Yes.
Yeah. Yeah. I think that's partly right. I think, but it feels different, right? Like, and so that was part of the question too, or like, right? Is it a portrait of that particular person and like their microbiome? Or again, does it speak to something sort of deeper? They're like, I think actually there's a really close friend of mine is professor at Tufts named Ben Wolf. And you should look up his research. He does like a lot of cheese microbiology. And so he sort of has studied like more closely the species. And so he was telling me more again, like they might be the same genus, but different species. So like they're closely related, they're cousins. And so like there's some people sort of speculate, like maybe, we touch the milk when we make the cheese, like maybe there's some connection, like, or early days, like that kind of connected our bodies and the food more closely.
Puts a new spin on handmade cheese.
Right?
Yeah.
Maine.
Sorry. I think there's a little bit.
Hi there. I think this question's for Scooby. I'm curious, first, just where electricity is coming from and going to in a plate. Is it like the root talking to the top, or the top sending back something to the root? And when you are listening to the electricity, your phrase, of how much is that a function of the location or other factors? You might be hearing something that's such a connoisseur that you're like, that's alewife, I know it. So I pull up, it is somewhere in alewife. Or is it more like, that's a chestnut tree, and I would hear that the same in alewife as I would in Lexington, as I would in the Middle East?
Hello. Good question. The system, I'd say it's not as accurate to know that like I can tell the species of like the trees. I guess that's for me. There's enough science to support the idea, but I'm really interested more in the artistry, like collecting all these recordings and then like sharing them with people, and also the electricity question. So it's really like the whole system. But I mean, even if you change the location of where the electrodes are, like tonight I shifted the location in the bromelia. Like most of the time they're next to each other on one leaf, and then I kind of spread them across from one another. And so the reading kind of like shifted and like kind of got a little slower because like that micro voltage that it allows the system to work, it took longer to get through the plant. And also if the leaf is dying, then like there's like that resistance, it takes longer for it to get through the cells to be able, for the measurements to be read. I mean, water is a big factor. Like I've done it, like that copper beech tree that was in the slides. There was a week that it was silent, and I thought there was something technical that was wrong. Like, oh, like the other electrodes came off the leaf or the leaf tore. But I decided just to hold out, and I was in my studio, and I heard a thunderstorm coming through, and I waited, and so within like an hour, the Copper Beach started live streaming again, and it just and I'd looked up the weather data, and there was like a mini drought in Cambridge at that time, so the tree was just very very thirsty and in a drought state, so it wasn't producing enough electricity for my system to read. So within like that storm passing through, then everything came back, it came alive again.
That makes me
Yeah.
wonder, thinking of the communication, um, do you go into the roots or consider where they, are tapped into the mycelial network?
So I've actually never connected to the mycelial network. I think I have to shift some things. Like I'd really need like very small electrodes. But I was doing a demo like a year ago, and one of the questions during the Q&A was if I connected two of the plants that were potted, like if I could get a reading from the two. And I'm like, it was actually like a young boy asked that question. I'm like, oh, what do you think? He's like, I don't think so. And I'm like, I don't think so either. Let's try it. So we did it, and I explained it like the circuit can't connect because the plants, like nothing's touching. So we tried it outside with a tree. I think it was like a maple tree, and then there was like a shrub maybe 20 feet away. I'm like, well, let's see. Maybe maybe the roots are touching and talking to each other. So we tried that, and sure enough, it started generating music. The changes, kind of the contour of the music, it was much slower because like that kind of that micro current, it just took longer to get through the pathway, but it definitely it showed that they were like above ground, they were not touching, and they were like far away, but underground, they were holding hands. So
That's beautiful.
yeah.
It really highlights how it's an art of listening more than making in your version of sound music. Yes. Oh, we have lots of questions.
Hey. I know you mentioned something about bacteria and the body odor and all that production. But I've heard that reasons why mosquitoes bite or sting some people more than others is based on certain blood types. Is there any truth to that?
So I'm glad you asked, because that is probably like the most commonly believed reason, but it is not actually true. So blood type, or blood sugar, blood sweetness, people often say, does not have to do with mosquito attraction. There is a very real difference among people. Like some people are very attractive to mosquitoes, some people are not very attractive, and everything in between exists also. And you can see it in the lab, but there isn't anything we know of in the blood itself, which, right, if you think about it, like the blood should be on the inside of your body. And on the outside are things that are making the smells. But it also, like, yeah, I think people have really looked and really wanted there to be some sort of a biomarker in the blood, but so far there's nothing that we know of.
And I know that scent and taste are so closely linked, but are you saying that they don't taste sweet blood at all? So we understand it is just the smell.
Yeah, so they use smell to find people. Once a mosquito starts to drink, once it probes,
Yes, familiar.
it's this really disgusting process where it's like looking for a vein. It actually doesn't need to be any sugar.
No
And
taste.
you shouldn't have everyone should have the same amount of sugar in your blood. And if you don't, go to the doctor. They actually use this kind of generic signal ATP, which is it's basically what all cells use for energy. And so it's super generic, not just among humans, among basically all life. And people have been very surprised to find out that once you get them to start drinking, all they need is ATP as a signal to keep drinking. And they'll happily consume a meal full of ATP in like a saline and then realize they can't mature their eggs and evacuate all of it and start.
I love the
But
science
they-
speak. They evacuated. Next question.
Question from Beck. For vision, for sound, for touch, there's a lot of mechanical sensors or digital sensors that we use every day. Is there a difficulty of doing the same thing for olfactory sensors, or is there any other reason?
Yeah.
I don't think they exist. Right?
This is the best question because this is the question that's like nearest to my heart. So smell is really complicated, right? Anything that is a chemical that's volatile could be a smell. So like with vision, we know it's a spectrum and there's hue and intensity and saturation, and you could digitize it really easily. With smell, people don't know where to start. And I think we're at this moment where, you know, I think people are starting to work on this a lot, and hopefully that we will reach a place where we'll be able to digitize smell, but no one has ever come close.
For people who still want to go to grad school, that's a hot topic. Next. Let me see if I can grab one of these. Test. Great. Right, maybe we're there. Yeah.
Channel Usher.
Hi. I have a question for Jerry. I'm very curious about your work with mice, and particularly in relation to something Meg said earlier about chemosensation. What sort of senses are mice experiencing that we might not necessarily experience as humans through the fact that they have whiskers, and how does that change kind of what either like brain mapping or neural pathways like look like in a scan? Like, is it more spread out across their brain, or are they activating specific sensors the way that we would see in like an MRI scan? How does that differ?
Yeah, I think there's some differences. So one thing when you pointed out the whiskers, and I mentioned that they use their whiskers a lot for touch. So I think maybe you've seen like the homunculus map of the human brain and how certain parts of the human brain or parts of the body are more overrepresented. So you have very large lips representation or large fingertips. So if you look in a map of a mouse, you see a large whisker head. It's quite enormous, and right. And one of the reasons why we study whiskers is because we can easily find parts of the brain that responds to even a single whisker and start to record from it. So that's one big difference, or one thing that's accessible, and it's from from humans, from mice, and even other species of animals that have whiskers. They use whiskers, like seals, for example. They also have this large representation. And the other thing that's a little bit different, which might be it's kind of a difference between small brains and big brains, right? So we have a big brain, mice have small brains, and you can see that the way that the brains are organized could be proportional to the size of the brain, and that these areas are much more interconnected and less hierarchical. The human brain, things are much more partitioned into hierarchy. So there are some differences between species in terms of how the brain is also organized.
Great. Well, I think we have time for 2 more questions. We're going to wrap someone in the back.
Yeah. All right. So this question is on. This is on. Is it working? Yep. Okay. This question is for Scooby. We have had students now work with the smaller versions of these, the plant waves, and doing this stuff. And they had first made the assumption also that it would be photosynthesis that would create this effect in terms of creative music. They actually did some studies looking at damaged leaves, sun leaves, shade leaves with oak trees, which do this. My question, you brought up water, and that was their next natural question. They said, well, what about water? Is it just light that's causing this, or is it water? And I don't know if you're familiar with the work of Vita Austin, who's been doing a lot of stuff called hydroglyphics, where she's taking the fourth phase of water and then being able to kind of see this symbolically. So they've taken it to the next step now, where aside from the music, they're actually playing the music from the tree to the water. And then the water they freeze it just like for 5 minutes, and it creates a hydroglyph. So it basically can say, in essence, am I receiving something happy or am I receiving something sad? The same way, I'm sure you're familiar when they used to go in and like yell at a tree and tell bad things to it and it would start to die. You'd see it after a while versus flourishing. She's able to capture this in water. So I'm wondering if this is anything that is of interest and exploration for you.
Thanks for sharing that. Actually, I didn't know about that work. Yeah, I'm always interested in, like, where I can take the plant music, like what's next. I know people, like the gentleman was asking about, like, can you tell, like, if this is a cedar tree, like in this part of the country versus another part. So I feel like starting to combine like more sensors, maybe to kind of like have just like have more data to, like, to kind of like qualify like what you're hearing. Like, I've noticed kind of big things because I've tried to record in as many climates as I can. But yeah, that sounds really interesting. Like, yeah, that work.
When you record in the desert, is it quieter out there?
Well, related to like photosynthesis, like I've done it when I first recorded, like when I first started the project, I was in Joshua Tree, and this is like a musical project with a friend who's a guitarist, and so he's improvising while we're listening, and we noticed when it was getting close to sunset that the data was kind of spiking, like the plants were getting more active, and just because we didn't know, we're like, well, the sun is setting. Why are they kind of coming alive? And then we realized, like, in the desert, that's what happens, right? They're going to open their stomata when, so that they're not going to burn all the moisture off. So we could see that in the musical data that we were capturing, that like we saw these spikes as the sun was setting.
They're getting ready to receive the dew and the coolness from the night.
Yeah.
Wow,
Yep.
That's great. And I think we have Amber in the back looking for another question.
Is it working? I don't think so. It does not. Okay, I just can't hear myself. Hi, this was a question for everyone. Despite the differences in the structures or the brain that you are studying, do you guys have any recommendations of sensory experiences that we as like humans could incorporate into our everyday to benefit us?
Something of the sensory experience. Extra bonus points if it is not in your specific domain and why you were here tonight.
Yeah. Obviously, the first thing that came to mind is to smell some smells, but I think it's really whatever you want, whatever area of your life you want to enrich. So, if you start to smell different fragrances or feel different textures and make it a part of your routine, you're going to expand that area of your own experience, and you'll see those changes, and it's pretty amazing. So I think you kind of can't go wrong.
Christina, you have a very mischievous smile.
I was remembering, like, maybe just like jumping off of that, like I didn't like smells before I started working on this project, and I didn't like eating cheese, and I thought it was gross and bad. I didn't, yeah, like, and I was like, oh, gross smells, and so actually, like, I think, yeah, kind of purposefully like experiencing something and trying it many times and understanding to sort of pick apart the differences and the nuances and thinking about them, like actually now I really like Right. And so I think that that's something that's quite kind of interesting. It's also something that works for little kids. You have it, about the tiny tastes. Like if you actually
Tiny taste?
tried, you try something like 5 times, like with just a tiny taste, you actually start to like it. So like even things again like that you think you don't like or you think you're not attracted to, you can learn you have this kind of fussy experience, which I think is cool.
And Jerry, the plasticity, thinking about what you're coming up with in your lab and how you're trying to um, expand the capacity, which is what we're wondering about.
Yeah, I mean, this is actually something that I studied during my PhD, was kind of comparing, when you're younger, right, when you're growing up, your brain is more plastic. That as you get older, for whatever reason, becomes harder to pick up new languages and stuff like that. And there's a lot of things that you can do to kind of train your brain or expose yourself in a way where it kind of reactivates this kind of more juvenile state of plasticity. And so that's a very heavily active area of research to kind of, reactivate these juvenile states of plasticity.
So
You
be
can't say that we're just giving us any of that. Give us one. What's the thing you do?
Well, one of the things that we studied, which is a little bit kind of circuitous, was Prozac, actually. So, so we were studying, how, what can you do to reactivate the juvenile state, and there was some studies that were kind of suggesting, oh, maybe serotonin reuptake inhibitors, which is Prozac, which is usually used as antidepressants, actually can reactivate these kind of juvenile states of plasticity. I don't know if people take Prozac for that purpose, but it's one of these, it taps into some of these related pathways.
So we can try new foods, little tiny tastes, and take rest. Scuba, do you have a sensory thing you wanted to add?
Just with your talking about plasticity, like with the plants, you essentially whenever I'm recording or performing with them, it's all improvisation. So I feel like that relates to plasticity. I just have to listen and be in tune with what I'm hearing, because it's never the same. So I'm if I'm accompanying the plants, I've just got to be present and in this listen
and tuning in.
Yeah.
I think that's unfortunately a great note to end on for tonight. I love the tuning into our senses. Thank you so much, Christina, Meg, Jerry, and Scooby, and all of you for being here tonight in this cold winter weather, and most of all for Decordova for being incredible for hosting us tonight, opening up this beautiful space. From everything from the chairs to the AV, they're wonderful. And I hope you guys take a moment to appreciate the art here. Thank you so much to Cordova. And subscribe, fill out our survey, and the cookies are on us.
There should be another slide with a survey.
Oh, let me see. There we go. Thanks so much, everyone. We'll see you at the next one. Good night.